All right. My name's Viny. I'll be presenting some early work on behalf of the Ross Levine Lab, where we're using this technology to help us evaluate minimal residual disease monitoring after therapy and clonal hematopoiesis. And really a critical part of that is a really precise understanding of the clonal architecture as the disease progresses. And so it's not lost on this audience that in myeloid disease in particular, the clonality changes as the disease course changes, and it changes in response to therapy. And thus, in a patient who has a myelodysplastic syndrome and thus a clonal abnormal bone marrow, further acquisition of additional clones is going to alter the clonal landscape in the bone marrow and lead towards selection and polarization at the time of leukemic transformation. These analyses have in large part been done using algorithmic assumptions. And this data here is in work from our lab where we're using bulk sequencing, using these algorithms to derive the clonal architecture of these patients' disease. This, of course, is done on bulk sequencing and thus is very good at resolving the dominant clones. But I think, as we'll see, and hopefully as I'll show you, that is not seeing the entire picture and may miss very key important parts of the patient's disease and therapeutic tractability. And so, in addition to the architecture itself, the clonal acquisition of mutations also matters and also affects prognosis. This was a figure that we put together actually in response to the work in New England Journal of Medicine that Kathy had just presented. And in fact, the acquisition of these additional clones, these additional mutations, changes the natural history of the disease. There are, of course, certain leukemias where the genome is relatively silent. There's relatively a paucity of mutations, such as the, I guess we're not getting any laser pointer there. There we go. Like a biallelic CBP alpha mutation, which is going to lend towards a leukemia that happens to be relatively chemosensitive and relatively silent from the genomic perspective. Whereas there's other mutations, such as chromatin spliceosome mutations, that acquire additional mutations. Some with p53 mutations, through instability of the genome, are going to acquire new mutations that are going to create a very complex architecture and are going to affect the way that we should think about treating these patients. And so, in addition to the technologies that have been developed to identify these mutations, these technologies are also used to monitor the clinical response. And so, historically, for the last 40 years, we've used a cutoff of 5% bone marrow blasts by immunohistochemistry as our definition of complete remission. Of course, now in the era of next-gen sequencing, RT-PCR for certain fusion proteins, and flow cytometry with 10-color lasers, we now are able to determine the depth of response far beyond the depth that we can appreciate with immunohistochemistry. And so with these technologies, we can roughly get to about 10 to the minus fourth depth with next-gen sequencing, of farther depth with RT-PCR. But the point is, this is actually clinically relevant. And so if you look at patients who have a greater than 5 log fold reduction in their NPM1C mutation after chemotherapy, these patients inherently do better. So the depth of response actually has prognostic value. So really understanding the clonal architecture to a degree of 10 to the 4th or 10 to the 6th is going to affect whether or not this patient requires additional therapy, whether they can be monitored, or whether they need therapeutic dose intensification with a consolidation strategy like an allogeneic stem cell transplant in order to try to get this patient into a real bona fide cure and remission. But intensification strategies don't discriminate the specific disease alleles. They're just determining whether or not there is some clonal marker of persistent disease. And so, as Tim Lay's group has shown for years, the clonal architecture is actually extraordinarily complex. And knowing which clone is the clone that's going to grow out is really going to matter on whether or not your therapeutic strategy is going to be effective. And really inform you, especially as new therapeutic tools are coming out, which strategies should be employed. And so we embarked on a proof of principle study with Mission Bio and took 2 samples treated at Memorial Sloan Kettering Cancer Center. We took FACS-sorted CD34 positive cells in 34 positive leukemias. And we took patients whom, between the 2 of them, had 5 identified mutations across 4 genes by our CLIA assays. We ran this on the Mission Bio AML panel with 19 genes covered, 50 amplicons, and 1 MiSeq run per sample. The first patient that I'm going to present is a patient who had AML, received induction chemotherapy, and had persistent disease at their day 28 bone marrow. This patient had acquisition of 2 additional mutations not identified on their diagnostic specimen, and actually contraction of 1 mutation after induction chemotherapy. The second patient I'm going to present is actually a patient with low-risk MDS, has been followed for about 2 years, had 1 bona fide driver mutation identified at the diagnostic specimen, had been followed with transfusional support and growth factors for about 2 years, at which point the patient started needing transfusions more frequently. A bone marrow biopsy was performed, and we found 5 new mutations on the background of the driver mutation originally identified at their diagnosis. And I thought that the 2 patients presented here are really illustrative about the power of what we can learn from this Mission biotechnology. So these are the summary statistics just to show you the input of cells, the number of cells that we can sequence by single cell, the number of reads, the on-target reads, and allele dropout rate. And so with one of these samples, we had excellent cellular input. For patient 1, we had 350,000 cells that went in. We were able to sequence 7,100 individual single cells with read counts of on-target reads over 80% and allelic dropout rate that was acceptable. The second patient, unfortunately, we weren't able to sort as many cells as we had hoped. And so we only were able to get about 100,000 cells. But we still were able to get over 15 million reads and sequence over 1,000 single cells. So even with suboptimal input, the real world application of this technology really is still going to give us some really exciting, really important data. So here's the results from patient 1. Patient 1 had originally been diagnosed with a leukemia that had 2 mutations. One of those mutations was lost, and 2 others were new at the second bone marrow biopsy. Actually, all 4 mutations were identified in this specimen. So although our CLIA assay was not able to detect one of these alleles, one of these mutations, the Mission Bio single cell was able to detect it. And actually, really surprisingly and quite strikingly, this is 4 cells. In 7,100 cells, they identify 4 cells that are actually persistent mutations from this patient's original diagnostic marrow. Obviously has contracted in the setting of induction chemotherapy. This patient got further chemotherapy, was able to achieve a complete remission by immunohistochemistry with MRD positivity, and ultimately, unfortunately, relapsed. When she relapsed with this clone. So this is a clinically significant clone that, although it was thought to be eradicated by a CLIA assay, we just couldn't appreciate it because the small size of the clone. At this patient's bone marrow in particular, we found that the dominant clones were actually these
these triple mutants that represented about 90% of the variant allele frequency, about 90% of the clonal architecture. And the clone that was the most virulent, the clone that ultimately this patient relapsed with, represented only 0.07% of their tumor. I mean, it's just shocking the power and depth that this technology is able to detect. And so just to reiterate here, in addition to the fact that we detected this extraordinarily small clone, just as Kathy's data showed, our variant allele frequency otherwise in the large clones really matched up quite well with our CLIA assay, with roughly no difference greater than 10% in the clonal architecture of the large clones, just suggesting that these algorithmic assumptions we make are very good and generally appropriate for dominant large clones, but we just can't appreciate small clones that are below a 10 or 5% variant allele frequency. And in this fact, actually ultra low variant allele frequency. Patient 2 was the patient with low-risk myelodysplastic syndrome who is now transformed to a more aggressive disease. And this is actually before this patient ultimately transformed to AML. But you can see here that the clonal architecture is actually much more heterogeneous, with really 3 clones representing roughly equal components of this patient's disease. But what we think is actually really surprising here is we could not appreciate through bulk sequencing that one of these 3 clones actually has homozygosed the original driver mutation. And so although all of the mutations were detected by a CLIA assay, all of the variant allele frequencies for the individual mutations were roughly identical, the copy number and zygosity of these mutations absolutely could not be. And so while we identify a particular variant as a large double mutant clone, it has a completely unappreciated homozygosity of a driver mutation that was unable to be appreciated by bulk sequencing. And so between this patient's variant, which again matches up very nicely with our CLIA assay in the architecture of 3 relatively large clones, the copy number of these mutations, the zygosity of these mutations just simply is not able to be appreciated by bulk sequencing. And so we were able to then follow this patient to determine if their further disease it represents a homozygous leukemia. Those data we'll have hopefully in shorthand. So in summary, this is an assay which is extraordinarily user-friendly, and even with cell inputs that don't meet the minimum recommended input, we're still getting excellent reads, we're still getting good coverage, and we still can get a very fine resolution of single-cell sequencing in a heterogeneous patient sample. We had low allele dropout, we had very good pi panel uniformity, but what's really, I think, powerful is that compared to bulk sequencing, we have an ability to detect small clones that are clinically relevant. We have the ability to determine zygosity in the mutant clones, which is really essential for appreciating the clonal polarity as these diseases natural history changes. But we were able to detect a clone that exists in 4 to 5 cells of a patient's leukemia, a 0.07% clone that really has tremendous clinical relevance in this particular case. So we were joking when we looked over this data that both we and Mission Bio got very lucky in selecting these patients because this data is just, it's just killer. So we also had a very strong correlation between our variant allele frequencies and with what the Tapestri was able to detect. I think really just confirming that bulk sequencing is really quite adept to identifying large dominant clones. But if you were in Irv Weissman's talk yesterday, he charges that we spend too much money studying bulk tumor and not really understanding the drivers of these leukemias. And so if we want to get at the roots of treating leukemia, I think this is the technology that's really going to help us do that. And I'll be happy to take any questions.
Can I ask you, how do you see a progression to solid tumor diagnostics, if you see it?
The question was whether there's an application for solid tumor diagnostics, and I'm a leukemia physician. I see leukemia patients, and I'm a bit biased in thinking that we, in the hematopoietic oncology field, are very far ahead of our solid tumor colleagues, which is only a half truth. But in all honesty, I think the clonal heterogeneity of solid tumors is increasingly not only being appreciated, but being studied and explored, and the metastatic initiating cell and targeting those cells is really where I think the field is moving in the world of solid tumor. I was excited to hear Dennis mention that the paraffin-embedded tissue assays were quite robust for prostate and breast and some other solid tumors, so I think that would be interesting.
Collection. But logistics of the sample collection in the duration of the treatment and all that.
Yeah, I mean,
you guys are getting it very easy.
Yeah, the question was the logistics. I agree, and as a simple-minded blood person, I get my tumor biopsy with a butterfly needle, and so it's quite easy. So I'll see if you have any, Dennis, if you have any other comments to the logistics.
Yeah, I don't have too much to add to that. I mean, I think it's there's definitely questions about where you sample the tumor, and are you appropriately representing all portions of the tumor, and so there's some considerations there that need to be given. But we have a robust assay working for fresh frozen tissue. So if that's the type of sample you can acquire, I think it's very amenable to our type of analysis.